the central dogma of biology. The central dogma of biology is one of the fundamental concepts in molecular biology. And the term is credited to Francis Crick in September 1957, where he referred to this through a text written saying, once information has got into a protein, it cannot get out again. So, central dogma of biology. Now, maybe some of you already heard about the central dogma of biology, and you know it is all about three terms: replication, transcription, and translation. However, I would like to give you an analogy to this story. Now, this would take me to some old memories where when my mother was telling me that when she got married and she was flying to meet my father on her way from her hometown to the airport, during that period of roughly 2 to 3 hours, my grandmother was giving her recipes. So there was a recipes book, okay? That's the recipes book that my mother had written during these 2 hours. So she was writing very quickly these notes, okay? So this is the recipes reference book, okay? And you can imagine that this, the way I learned about this is that after many years, and I guess when I was maybe 9 or 10 years, I saw this in one of the drawers, and I liked it because it had the recipe of a cake, how to prepare a homemade cake, and you know, I had all the details there. So because it's such a valuable thing, I had to go and write down, take notes of the recipe on a separate piece of paper. So let me change the color here on that separate piece of paper. So I had to transcribe, okay? I had to transcribe that specific recipe and keep this protected, okay? Eventually, I was able to prepare my cake, okay? My cake. And that is meant that I was able to translate the recipe to an actual outcome and product. So in similar analogy, we can think about the recipe book, the reference book, as the DNA, okay? And the transcription is the process that we go through to take the DNA to RNA, and then from RNA, we translate that into proteins, okay? That is, in a nutshell, the analogy that we can draw here, okay? Now, when we speak about central dogma, there are three main processes, again, that we want to refer to. There's DNA replication, okay? DNA replication. And DNA replication means that DNA can make a copy. DNA make a copy of itself, okay? The DNA can make a copy of itself, and that is very important for, as you would imagine, when we described this earlier, for specifically cell division, okay? Cell division, and to be able to replicate the information that we have in one cell, okay? And that start producing the, information needed for the other cell, okay? And excuse me, another important process, which is part of, as I mentioned earlier, is transcription, okay? Which means we're gonna read a specific region or segment of the DNA, for example, a gene that will be used as a template, okay? that will be used as a template to synthesize the corresponding information in a shape of what is called RNA, ribonucleic acid, and we already referred to this earlier. If we get this gene and we get the information eventually, the RNA here, we usually call, or we actually call this as mRNA, messenger RNA, which means the RNA that comes from the gene source. Now, once we have the RNA, there's another important stage, part of the central dogma, okay? mRNA itself now is going to be a template. It's another template, and maybe I draw this that way. So gene is a template to get RNA, and then, or mRNA, and now mRNA itself is a template to get what? The protein. Okay? And this is usually happens through the process where there's what's called a ribosome kind of proteins that read the sequence of the mRNA and start assembling information into what is called amino acids. So these proteins are made of amino acids. We'll see this in the coming slides. Okay. So how can we summarize the central dogma of biology? As you would see here, we can summarize it as DNA transition to RNA, which we call transcription. And this is single-stranded, not double helix, as we have the DNA, and eventually protein, right, which is a complex chemical structure. Okay, that is how we can summarize the central dogma. In other words, DNA codes for RNA, and RNA codes for proteins. Okay. Now, in general, that is the flow of how genetic information goes from DNA, genetic information, okay? This is, in general, the direction of genetic information. One of the exceptions for this, for example, is that the process can go backward from RNA to DNA, something that we can point at as reverse transcription. Okay, reverse transcription. Now, this is an interesting term to point out. Since reverse transcription is usually used as a mechanism in some viruses that they don't have DNA and they have RNA, and still they won't replicate information from that RNA. Yet in research and biotechnology, there's a very important use for this reverse transcription idea, which means during a laboratory setup, we need this idea of reverse transcription, which is essential, as I said, in the research and molecular biology research and biotechnology, to achieve a certain important goal, and that is directly related to gene expression. Okay, gene expression. Something that we will revisit through the course as we learn about PCR technology, as we learn about microarrays and gene expression. Some of the concepts that we will come to. For example, in reverse transcription in the PCR, or what we can refer to as RT-PCR, polymerase chain reaction, okay, is important in detecting and quantifying the presence of the specific RNA molecules, which eventually is going to tell us the level of presence of a specific gene, okay? The level of presence of specific genes. So this really relates to RNA because DNA will never change in terms of the quantity. It is the reference book. But how many times I cook the recipe of my grandmother, I take and transcribe it. I can transcribe it into many, many times, as I did in my marriage. I keep going to that book. Every now and then it's ripped off, and then I go back and do it again. And maybe sometimes I do multiple copies, give it to other family members. As many times, the reference all book is still as is. However, the times we can transcribe that can be many times. And maybe I've been referencing a specific recipe, not all recipes as frequently as such. So in this case, this is directly related to gene expression. And so central dogma of biology, as you see here, goes in these two directions with three important processes: replication, transcription, and translation. And we also pointed out the idea of reverse transcription as we see here. Okay. Now, we want to dive into a very interesting step, okay, which is actually the first concept in the central dogma of biology, this fundamental concepts in molecular biology, okay? That is DNA replication. Now, this might be, as the viewer here might seem, you know, it's very interesting drawing with lots of keywords here, and that's why what I'm going to do is I'm going to draw some sketch, simplify it, and then we'll end up with this overall view, okay? So let me do this here. Sorry. Oops. Okay, let me put a dark screen here. Very nice. And then what we're gonna do is look into something interesting in terms of DNA strands. Now, just I believe something that I skipped without referring to is, let me take you to this slide here. You see these backbone sugar groups, right? They had 5 carbons. So this is what we call 1', 2', 3' is here as written, 4' is here, and this is the 5th prime. Okay, this is 4', and this is 5th prime. So this one is the 5th prime. Okay, so as you see, at this 5th prime, the phosphate group would come, and then the next one would link to the 3rd prime and keep going. In that way, they were able to explore and define a directionality for one strand that is going from 5' to 3', and the opposite strand is going from 5' to 3'. Okay, so in other words, if I come back here to this slide, what I want to sketch for you out of simplicity, okay, is the following. Imagine that we have a circular DNA. Now, this can be the shape of DNA in maybe in some viruses, not in the human. We know we don't have like a circular DNA, but just, you know, as for the purpose of illustration. So we have one strand going in this direction, and that is 5' to 3'. Okay, and let me switch the color here. We're going to have another one. I'm going to put some distance here. Okay, very nice. And this is going to be in what direction? In the opposite direction. In the opposite direction. So 5' would be here and 3' would be here. And again, this directionality is just a reference of how the molecules are arranged, and specifically when we look on, you know, one side, we start with one side, end with the other one, we see that the linking going from the 5th carbon ending by the 3rd carbon, and so we use this as a reference to know that, oh, one strand is opposite to the 2nd strand. One strand is opposite to the 2nd strand. So here's an exercise. We know that A, C, G, T, T, for example. What is the complement for this? What is the complement for this? Another question would be, what is the reverse complement for this? So I want you to think for a moment. And we have done this exercise earlier, where we have derived the complement, okay, where we assume it's going in the same direction after unfolding and having two strands. But actually, this is going in an opposite direction. So if I were to ask you the reverse strand, I want you to be careful in how you derive it, okay? So I'm going to give you a moment. And again, take the chance to note this question down and give me your answer to confirm that we are watching the lecture, okay? Great. So the reverse complement of this, you may start with saying, well, A pair with T, G, C, A, T, A. But the way we write the reverse complement, because this is going in the reverse opposite direction, is going to be actually opposite. So it's AT, A, C, G, T. Okay? And that is the reverse. If I want to write a function saying reverse complement, okay, A, C, G, T, T, A, T, that is going to be the answer for that reverse complement, okay? So just to make sure you capture this idea, okay, great. So now I want to go back to the two strands of this DNA and go work with this directionality that we have in front of us. That is key. This directionality is key for knowing how the DNA replication works, for how the DNA replication works. Let me use this color here, okay? So now as we look into this, imagine that this is the what is referred to as the origin of replication, which means it is the site where the replication process would start. So let's unwind the two strands and start to stretch them. Okay, so we're going to stretch this to be a little bit like this. Okay, and then we're going to stretch the other one, cool, to be a little bit like this. Okay, now this is stretched. Now, if I were to ask another question that I want you to note down and answer, right? Knowing that we want to do this process in a very efficient way, right? How many forks we can do in parallel to do replication? As we go and we unwind and fold this, and what are the possibilities to make this a very efficient replication process? We know that once we stretch this, we are trying to get some information and generate the complementary here. And the other one, we want to generate some information and come up with the complementary here and eventually take the new information. So as we unwind this, maybe there's some shape here proposing certain mechanism of copy, okay? And here it's worth to mention what is called DNA polymerase, okay? Okay. Very slowly. So DNA polymerase read the original strands and adds the matching nucleotides because it is the enzyme responsible for this process. As long as RNA polymerase, which we're going to see soon, is responsible for reading and copying information from RNA. So now going back to the DNA polymerase, let's assume we have 4 DNA polymerases. We can think that we're going to stand here and fork one process in that direction, fork another process in that direction, and the third process in that direction, and the fourth process in that direction. And here you go, in parallel, an efficient setup for this is to go left and right, and that's it. That is great. And you are trying to cover all the DNA, and then you are replicating the the DNA in an efficient process. However, that is not correct. That is a wrong answer. Okay. The reason for this is that DNA polymerase, and this is a very important piece of information, are unidirectional. Okay. Unidirectional. Okay. Unidirectional. What does this mean, unidirectional? Okay. It means that it can traverse, let me write this for you here, traverse the parent strand. It can traverse the parent strand in one specific direction, that is 3' to 5', creating a new strand going from the 5' to the 3' direction. So DNA polymerase, when it's going to read the parent strand, it can work with only one specific direction, which means we have to visit how we view this part, okay? And so I'm going to introduce here a new color for us. Now, when these two DNA strands are forked, we can see that here this area is clearly going on the three, like it's an open area, which is easy for the DNA polymerase to come and work with and start generating information here. In the same manner, it's the same thing for the opposite strand here. So these two can work in parallel and smoothly, which means the DNA polymerase can deal with this area. Of course, it can deal with this area, and it is unidirectional, and so of course it can go that way. Okay. Now the question is about this region. Okay, this region. Okay, how do we deal with this region? And I think now maybe this got a little bit confusing with all of these drawings. So I'm going to take a quick moment here and bear with me, because this is a very interesting way to look into the DNA and understand a couple of important concepts or refresh with them. Okay, so we have here another area, and actually, yes, okay, not maybe very expert in drawings, but we are back here to say this is very efficient because we have the directionality defined as preferred by the DNA polymerase, okay, as preferred by the DNA polymerase. So this is the opposite one in this case. Now what you see here, well, this is where DNA is going to bind here, DNA polymerase, and bind here. Now, to be able to copy this part, it wants to bind here. However, this is a very tight area. So what happens is that once this starts unwinding, it's going to take this area and generate the replicated information. Okay, the replicated information. So let me draw this in that way. Okay, let me switch the colors here and use this for replicated information. So since this unwinds a little bit slower than the original one, so it's able to only read and generate a copy of only a part of this. And then once it unwinds, once it stretches more, okay, so here we generate one and another one, okay, and then in the opposite way as well, we generate one and another one. So we keep generating these fragments that are referred to as the Okazaki fragments. Okay, Okazaki fragments. So that is the process. What is important here is just to know the DNA directionality, and that in DNA replication, this directionality is really taken into consideration. And as we go through this process of DNA replication, there's what is called the leading strand because it's efficient and already there is a good binding location for the DNA polymerase. However, there is the lagging strand, which needs some stretching before this start happening, and then here, and then here, and then gradually here, okay? It cannot directly bind here. It needs some space. It needs some floor to jump in and work with. So though, as you see, this is not a perfect sketch, but maybe this is now the time to go back to this very nice schematic of the DNA replication process, which I think really worth spending some time and with. So, oops. Okay, so as we visit these concepts of the DNA replication, there's the concept of helicase or the DNA helicase, and you see the DNA helicase is in this area. Okay, DNA helicase unzips the DNA because it is able to break down the hydrogen bonds. You see, it's able to break down these hydrogen bonds. So it has an important role as try to generate the forking area for replication. Okay, and you can see this is kind of a shape of a fork. Okay, this is the forking area to start the replication in parallel, and we know we have to work on replication in a very efficient way to create many cells, right? So this is the helicase here. And then we talked about the direction of replication. Another concept that we refer to here is this is the DNA primase, okay? This is the DNA primase, okay? DNA primase make short RNA sequences that are called primers, okay, that are called primers, and they are the starting point for DNA. They are the starting point for DNA synthesis. To synthesize this DNA here, we need some starting point, okay? So primase is responsible to create, to make the short RNA sequences, the ones that you see here. So maybe I'm going to circle this. You can see this. So this is going to flag the beginning of some sequence as the replication process is happening. So these areas generated or RNA sequences are created by the DNA primase, which helps in the DNA synthesis. Okay. Now, DNA polymerase, of course, it's a very important protein here, right? It is actually responsible. Now we have several DNA polymerases, right? They are reading the original strands and adding the matching nucleotide. So you see here, this is the DNA polymerase. As it reads, you see the arrow here, as it reads, it's adding the matching one. And the same way as it reads here, it's adding the matching ones in this direction. Okay. Now there's, as the DNA unwinds, okay, you see this here, as the DNA unwinds, there's also a very interesting thing to look into here, which is called topoisomerase, okay, or DNA gyrase. It helps by reducing the tension of having the DNA, you know, getting twisted. We know there's a strong twisting there in the DNA as it is folded when we saw this earlier. However, right, this helps in reducing the tension of this twisting, the topoisomerase, okay? So once the DNA fragments, in this case, are made, these are DNA fragments, we know that there are these RNA regions created by the DNA primase. Once the fragments are made, oxonuclease removes the RNA primers. So these needs to be removed, and the DNA need to be filled with DNA and then glued together. So this brings us to DNA ligase, okay, which is this one here, okay. DNA ligase is going to be responsible to glue and connect specifically what? The Okazaki fragments. Let's assume this is one fragment, second, third fragment, the Okazaki fragments that are being generated. So I hope this gives you an overview of one very wonderful process in our biological system, which is DNA replication. And it is really core to protect the cells and generate information in new cells. A final important things that I want to also refer to is the DNA ends. The ending of the DNA is protected. So there is a telomerase enzyme which add what kind of protective caps. Okay, you can think that there is some cap here. This is the end. Okay, and some cap here. Okay, it looks at the end and add some caps as you have your drink and you want to cover it with some tip there and protected. Okay, and these caps are called telomeres. Okay, at the end of the DNA, and they're like, again, much like plastic tips that you can put to cover and conceal the end of the object. So as we move in the concepts of central dogma, we looked into the first process here, which is DNA replication in more in-depth. Something that we're going to look into here is transcription. So the first step is DNA replication. That's the first process. And the second process is transcription. And this is the typical process in prokaryotes. Okay, so DNA transcription in prokaryotes. This is the process in general when we refer to transcription. Okay, transcription. It is the process which where the genetic information and DNA, okay, is copied. The genetic information in DNA is copied to RNA molecule. Okay. So when again also dive a little bit and try to understand the process to appreciate the biology behind this and know levels of details that might be really important in as we analyze data. In many of the cases, it really makes sense to understand what is this DNA, what are these specific regions, what is the process. Okay, so when it comes to transcription, there are three main steps. Okay, the first step is initiation, and the second step is called elongation. Okay, and the third step is just termination. Okay, so let's look into these illustrations in the slide here, and you have some summary points about each. So this is the initiation step, step 1, and this step 2, elongation, and this is step 3, termination. Okay, so in the initiation, in this case, there is what's called RNA polymerase, which is responsible for synthesizing RNA. So this is the RNA polymerase, okay? And it binds the RNA polymerase. And one thing I want to actually tell you here, when we look into the DNA, it's a language again, okay? And there are specific regions that would mean bind here, is encoding a message saying bind here. Okay, like the origin of replication. It is that area that is saying bind here to start the replication. And the transcription process, it is the promoter area. Okay, and that is the area where the RNA polymerase can bind to. Okay, and promoter is actually a DNA sequence that signals the start of a gene. So we have a gene, and that beginning of the gene or upstream of that gene, there is what's called the promoter, and then RNA polymerase is able to start here, and this is coding start, okay, and start reading the information here and to transcribe. That's going to create what's called and form what's called the transcription bubble. Okay, you have here the transcription bubble. And basically the DNA double helix around the promoter unwinds, creating this bubble. So it means that we have the DNA unwinding here to give the chance for the RNA polymerase to interact. And you see here this reddish circle. This is actually a kind of transcription factors, general transcription factors that help with the process, that help the RNA polymerase with a process. Specifically in eukaryotes, transcription initiations require a set of transcription factors to assist the RNA polymerase, okay, and begin the transcription. So that is the beginning of the process. So beginning of RNA synthesis. So using one strand of this DNA, okay, using one strand of this DNA, which we call the template strand, to guide the generation of the genetic information. To the next step, we have RNA polymerase, which is going to add RNA nucleotides that will be complementary to the DNA sequence. So that is a summary of the first step, okay? And the second step is elongation. And elongation is mainly meaning that the synthesis continues. So the process starts with the initiation. We know that there's a specific region to bind to. And then RNA polymerase moves along the DNA, continuing to add, keep adding. As you get new nucleotides, try to bind them together. And there's a short area what that's called DNA RNA hybrid. And that is as RNA polymerase reads the DNA, a short area that is this RNA DNA hybrid is temporarily formed, okay? This area where they bind together is temporarily formed. And the RNA polymerase keeps moving. And a very natural question here would be when this is gonna stop, okay? And that takes us to the third step, which is termination. It is the end of it is the position where the RNA polymerase is able to recognize that there's no gene anymore. Okay. Now the exact mechanism of termination is more of an organism dependent, and it depends on several factors. Okay. For example, the end site in eukaryotes can be either 11 to 18 base pair sequence, and characterizing this can be differently in prokaryotes, and so on and so forth. But this keeps working until there is a sign where to stop, okay? And then the RNA polymerase is disassociated with the help, of course, some transcription factors, and then the RNA transcript is released. So now we have a reference RNA transcript, and there is a directionality as well for that. You see the 5' here and the 3' here, which is going to bring us to this question: what is the direction that RNA polymerase move through to transcribe mRNA? So I want you to think about it for a moment, okay? And maybe as a hint, I may refer you back to, remember again, when we talked about the DNA replication and DNA polymerase, okay? So RNA polymerase transcribes mRNA by reading the template strand of DNA in the 3' to 5' direction. That is the template strand, okay? So hopefully sometimes I myself get confused with this. So you have to think about two things. You have to think, as we talk about the DNA replication, the parent strand and the new strand. And here as well, you have to think about the new mRNA strands. So RNAP or RNA polymerase is able to read, it's unidirectional, so it's able to go through 3' to 5' of the template strand, and the generated a new RNA, or the synthesized new RNA strand, is going in the direction of 5' to 3'. Okay, and so this is going to ensure that mRNA is complementary to the template strand of DNA. This is going to ensure, as you see here, that this is complementary to the actual DNA that it was came from. Okay, nice. And as we discuss this process of RNA, reverse transcription comes into light again. And reverse transcription is naturally observed in some retroviruses like HIV. So they have a mechanism where they can integrate RNA genome into the host's DNA genome. So imagine this, right? So this is really kind of unbelievable. So HIV virus is having this mechanism where they can integrate their own RNA. They don't have a DNA genome, yet because of their capability of reverse transcription, they can impact the host's DNA genome. Okay. So what we see here, it was referred to as oligo DT primer. And I think this is the main concept here in this slide. Okay. In a laboratory setting, reverse transcription is used, as I mentioned earlier, in specific technologies to be able to quantify gene expression. And that is through changing mRNA, because mRNA are these copies of the original recipe. So the quantity of mRNA, usually in the lab, they try to take the sample and isolate the mRNA, because this is what's going to tell us how much of that gene was there. However, mRNA is not very stable. And for the development of specific technologies, we need to convert this back to the DNA and to able to recognize which genes specifically they were responsible for this. That is why we, what is called complementary DNA is generated from this mRNA. Okay, a complementary DNA for quantifying and knowing the gene expression. Now, before I jump into oligo DT primer and the reverse transcription that are being developed and designed as primers to indicate the genes, the idea originates from understanding what is pulley A tail. Okay, pulley A tail. Okay, pulley A tail. Okay, in eukaryotic mRNAs, there's a specific characteristic attached to them. Okay, there's the polydenylated tail, that is the pulley tail at the 3' end, at the 3' end of eukaryotes. Okay, this is referred to as pulley A for short, and it is just a stretch. That's why they call it pulley A. It's just a stretch of adenine. It's just a stretch of adenine nucleotides, okay? Some group of adenines. That's why they call it poly A, okay? Now, oligo, and so again, this poly A comes, keep in mind, towards the 3' end prime, so towards the end site of mRNA or gene, okay? If we refer this, or sorry, the mRNA, this is towards the poly A tail is part of the end of mRNA. Okay, that's what we're talking about here as we look into this complementary DNA coming from the mRNA. So now, since we know what characterizes the end of mRNA, and we need to come up with an idea where there's a binding here. So we know A's or adenines, they link into thiamines. Okay, that is where oligo D T primer, T4 thiamine primer comes into play. Okay, and usually these are short sequences of thiamine nucleotides, something in between 12 to 20 bases long, 10 to 12 to 20 bases long. And since adenine pairs with thiamine, the oligo DT primer can specifically bind to the poly A of mRNA. And that's using this primer is going to ensure the reverse transcription, specifically targets mRNA for the cDNA synthesis, Okay, so once oligo DT primer is annealed to the mRNA, the reverse transcriptase can start synthesizing the cDNA. So you can think of this as we are having the mRNA here, and they were trying to come up with design that is the poly A, and then that's area. Okay, that we can establish some binding here and some reference to start a copying mechanism and generate the cDNA. A more stable version, the reference way of trying to quantify gene expression. And so here you can see some of that steps. And again, oligo DT would be the main concept in this slide. Another interesting concept to look into here is that there's what's called RNA-dependent RNA polymerase, RDRP for short. RNA-dependent RNA polymerase, which is an enzyme that catalyzes the synthesis of RNA from another RNA template. Okay, so think again about DNA polymerase was looking at the DNA, a template from the DNA. Okay, that is DNA polymerase we looked into this for what? For replication. Now we're looking at something else here. Okay, the RNA, the RDRP, that is the RNA-dependent RNA polymerase, is an enzyme that's going to be responsible again for the synthesis of RNA from RNA template. Okay, and again, one would start asking, is that possible? Is that possible to again synthesize RNA from the RNA template? Well, this is one mechanism that is found in RNA viruses, those that they don't have DNA. And so for them to survive and to keep their genetic material, they need to have to protect their RNAs, as we do for, or the process we do for DNA. They have to keep their RNAs through RDRP, and that is why these RNA-dependent RNA polymerases are important for protecting the RNA content and or genetic information. And as you see here, this is RNA genome for specific types of viruses that are called RNA. This is the viral RNA replication viruses. Okay. Now we come into the translation. Okay, so we went through DNA replication, transcription, looked into reverse transcription, and a little bit more details. But what about translation? Okay, so this is about coming up with again, we have lots of information, right? And now this is going to change to RNA. So in this case, you know, the complementary for this is going to be information going to be generated from this DNA. We're going to receive the mRNA, for example, if it is a gene. Okay. Now, this is going to go to be a protein. Okay. This is going to happen to be a protein as part of the translation process. Okay. Imagine that, you know, there's some secret message with some code there, and out of just possibility, imagine that there's AUT message, okay, that is saying, you know, hello for the sake of discussion, okay, and then another message is called AUG, okay. Well, I think just to make this simpler, because I cannot use T actually for the example I want to give. So I'm going to go back and say, imagine we have AUC, okay, and AUG. And both of these messages are saying hello. They mean hello for us, okay. What I'm trying to say here is there were interesting efforts by some scientists, okay, specifically here, Nuremberg, referencing back the translation process. For each 3 nucleotides, there's one associated amino acid that helps in constructing the protein. Okay, remember again, we have some RNA. Let me say RNA and then protein. Okay, so for each 3 nucleotides in the RNA message, they're going to code for 1 amino acid. So what are the possibilities here? We know that we have 4 for each 4 possibilities, right? AUCG, and then times 4 and times 4, that's going to equal to 64. So there are 64 combinations possible for that. However, Nuremberg was looked into this, okay, with joined with another scientist called Litter. They did an experiment called the poly-U experiment, okay, and in that experiment they made a string made up of uracil, which is part of the cell, and this string only produced one kind of information called phenylalanine, okay. Eventually, this helped in decoding the cell's language. So they were able to link this to 20, eventually to 20 different building blocks. All the combinations, possible combinations that they were all able to look into, they linked those into 20 main building blocks and only 20 different building blocks. Okay, so the building blocks that we have that reference the makeup of these amino acids is we have 20 of them. Okay, so this is usually the way that you can look into decoding and encoding that secret message, like the one I've been doing. AUC, as you saw in the slide earlier, this is AUC. So 3 of those nucleotides are going to encode for that specific amino acid, okay, for that specific amino acid. And what I want you to observe here is that AUC, AUC, AUU, AUA, so 3 of those, AUC, AUA, and the third one, AUU. All those 3 are going to link again to one of the 20 building blocks, okay? To one of the 20 building blocks that Nuremberg was able to find. So if you count those, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we'll go around this, so we'll find 20. So this outer circle is giving us 20 building blocks or 20 codons, and each of those is composed of 3, 1, 2, 3 nucleotides. However, and again, AUA and AUC and AU U are referring to the same codon. Okay, what could be the advantage of this? What could be the advantage of this is what you see here in the slide. It makes it fault tolerant. In other words, if forever there is some mutation giving any issue with the process and only the last alphabet or nucleotide flips, it is still we are able to decode the message and know that this is that specific amino acid needed for the creation of that protein, which tells us that the protein translation process is fault tolerant, given up that we have this detail here, this mechanism, and that we have 64 combinations, and those 64 link only to 20 codons again. It's not that AUU would give you something different than AUC. Somebody would say, well, that would have made sense. Well, it would have made sense in certain context. But here, for the sake of creating a protein, which is very important as for the biological system, some sorts of mutation ending up with maybe uracil instead of cytosine shouldn't change the output. Okay. And what we find here on the right side is I'm quickly going to refer to prokaryotic and eukaryotic mRNA process. Okay, and in this a process of translation of these mRNAs to proteins again, there's a ribosome here, one ribosome that we can see here, and this is in terms of measurement that can give us the dimensions of this. We have 30 S subunit and 50 S subunit in prokaryotic, and here it is 40 S subunits by 60 S subunit for eukaryotic. Okay. Now the main difference here in terms of the translation is there is a specific ribosome binding site to start the translation. So there is a specific location here that is not in prokaryotic. Okay, there's a specific location that is called Shine Dalgarno or SD. Okay, SD for short. And with regard to eukaryotic, okay, this is going to start here until it meets a specific sequence. Ribosome is going to scan actually instead of directly binding in this case. So the small ribosomal unit here is going to bounce at the 5, as we're used to the understanding this now, at the 5' and scans in that direction, 5' to 3' directions to locate that specific sequence here. You see here, oh sorry, the Cossack sequence, okay? The Cossack sequence, okay? And it's for my notes here. This is, I guess we can write it as ACCAUGG, okay, AUGG. So this is the Cossack sequence here. So it's another clear message that we're not able to understand it as a clear instructional English language yet. It is still saying something very important. It's here where the ribosomal subunit can start the translation or process of translation. With regard to this schematic here is referring to another concept that is called polycistronic mRNA. Okay, polycistronic mRNA. Okay, this is polycistronic mRNA, which consists of multiple cistrons, each of which can be translated into a protein independently. And so you see here is the same, and this is actually in prokaryotes. And so this is the binding site for the ribosome, and these proteins are different proteins, though they are coming from the single mRNA transcript. So a single mRNA script itself can be translated and produce different two or several different proteins. So this is concludes with looking as we look into the central dogma of biology, and I want to dive into the differences of between eukaryotes and prokaryotes, which we already started some discussion on. So we're going to dive into a little bit more details on this.